Calcium channel blockers, often abbreviated to CCBs, are also known as calcium antagonists. As their name suggests, they are a group of drugs that block the flow of calcium ions through calcium-selective ion channels. They specifically target voltage-gated calcium channels, often abbreviated as VGCC, although the channels can also be called voltage-dependent calcium channels and abbreviated as VDCC. A voltage-gated channel is a large protein molecule that spans the cell membrane and has a channel or pore running through its centre, which can open and close in response to a change in membrane potential. Cells at rest have a negative membrane potential, which means that the inside surface of the plasma membrane is more negatively charged than the outside. Also at rest, the concentration of free calcium ions inside a cell is kept very low, at nanomolar concentrations. As calcium ions in the extracellular space are present at over a millimolar, there is a steep calcium concentration gradient across the cell membrane, which favours calcium entry into the cell when a calcium channel opens. When the cell is stimulated, the membrane depolarizes. In other words, the inner membrane becomes more positively charged. This is sensed by the calcium channel, which undergoes a conformational change or change in shape, which results in opening of the channel pore. Calcium ions are now able to fit in the pore, and they can pass freely through the channel to the inside of the cell. Calcium channel blockers bind to a region of the calcium channel that prevents it from functioning normally. So when the cell is stimulated, it depolarizes as normal, but the calcium channel pore does not open. So calcium ions cannot pass through the channel to enter the cell. This blocks any signalling pathways that rely on calcium entry through voltage-gated channels for their activity. Due to their ability to prevent calcium entry into cells, calcium antagonists have a range of therapeutic applications. These conditions are all very different, and calcium antagonist drugs are not all equally effective in each one. So, let's look at which drugs are effective and the organs they target to bring about a beneficial effect. Supraventricular tachycardia and angina are conditions that affect the heart, and both conditions can be helped by drugs that act on the heart. A calcium antagonist that does this is verapamil. Like hypertension, angina can also be treated by dilating the systemic arteries. There is a group of calcium antagonists that do this selectively. They are known as the dihydopyridines. Another calcium antagonist used to treat angina and hypertension is diltiazem. It has both the vascular action of the dihydropyridines and the cardiac action of verapamil, although it is not as effective as verapamil against arrhythmias. Vascular spasm following aneurysmal subarachnoid hemorrhage involves the cerebral arteries, and one of the dihydropyridine drugs, known as nifedipine, has a selective action on the cerebral arteries. It is the only calcium antagonist used to treat this condition. The last category of therapeutic uses moves away from the cardiovascular system to the brain. Epilepsy and neuropathic pain are both neuronal disorders. Calcium channel blocking drugs that are effective in these conditions are the gabapentanoids, which selectively target the calcium channels in neurons. There is an obvious question: why do these drugs, which all act as calcium antagonists, target different organs to have these different therapeutic effects? In the rest of the video, I aim to give you the answer. To understand the differences among calcium channel blockers, you need to know a bit more about calcium channels. The main part of the channel is a membrane-spanning protein with extracellular and cytoplasmic domains that protrude on either side of the membrane. Inside the protein is a region known as the selectivity filter, which distinguishes between calcium and other ions, allowing only calcium ions to pass through. This protein, which is called the alpha-1 subunit, contains all the features needed to function as a calcium channel. However, it doesn't work alone. Calcium channels also contain a beta subunit attached at the cytoplasmic domain. They have a gamma subunit that is mainly located within the transmembrane domain, and an alpha-2 delta subunit that sticks out from the extracellular domain. These are all distinct proteins that have varying effects on the properties of the channel, including its sensitivity to drugs. Calcium channels are even more complex than that because there are multiple versions of each of the subunits. There are 10 different genes that encode for 10 distinct alpha-1 subunit proteins. 4 genes code for 4 different beta subunit proteins, and 8 genes code for 8 different gamma subunit proteins. The alpha-2 and delta proteins are the product of a single gene and are linked together by a disulfide bond to form a single functional subunit. There are 4 alpha-2 delta subunits encoded by 4 different genes. But the picture is further complicated by alternative splicing of each of the subunit genes, which leads to an even bigger range of subunit proteins. These are differentially expressed in different tissues. So, there is a lot of diversity in calcium channel subunit structure that could help to account for varying selectivity among the different calcium channel blocking drugs. Drugs can interfere with channel activity by interacting with different subunits. The neuron-selective gabapentanoids bind to a site in the alpha-2 delta subunit. This subunit plays a role in trafficking the channel to the plasma membrane, and the main effect of the drugs is to inhibit trafficking and reduce the density of channels expressed at the membrane. With fewer channels present, their activity is reduced. In contrast, the cardiovascular drugs, verapamil, dihydopyridines and diltiazem, all interact directly with the main alpha-1 subunit to reduce calcium ion flow through the channel. Among the 10 genes that code for the main alpha-1 subunits, there are 3 distinct families that give rise to calcium channels with distinct properties. They are CAV1, CAV2 and CAV3. The CAV1 family are known as L-type channels. The CAV2 family members are known as PQ, N and R types, and the CAV3 containing channels are known as T-type. These names came about before any calcium channel genes had been identified, so they reflect the biophysical properties of the channels, according to the ionic currents that they create when calcium ions flow through them. The names were introduced in this paper, published in 1985, when it was first realized that there is more than one calcium channel. One of the channels had a large conductance that lasted a long time, so it was called L. Another had a tiny conductance and activated only transiently when the cell depolarized. It was called T. A third type was found to be neither L nor T, and so it was called N. The PQ and R types were discovered later and assigned letters in between N and T. L-type channels are found in all muscle types, as well as endocrine cells, retinal cells, and some neuronal cell bodies. T-type channels are found in cardiac pacemaker cells, some smooth muscle and neurons. On the other hand, PQ, N and R-type channels are largely neuronal. When considering the cardiovascular calcium channel blockers, verapamil, dihydopyridines and diltiazem, only one of these alpha-1 subunits is relevant, the Cav 1.2 subunit, which is found in cardiac and smooth muscle. All three drug classes bind to this subunit, but they bind in different ways. These are the structures of verapamil and diltiazem and the general structure of the dihydropyridine group of drugs. They are quite different, so it's not surprising that the three types of drug bind to the alpha-1 subunit at three distinct sites. This symbol represents the heart-selective drug verapamil. It binds at a site inside the channel pore and enters it from the cytoplasmic side of the membrane. In this way, it acts as a direct pore blocker, physically impeding the passage of calcium ions through the pore. This symbol represents the vascular selective dihydropyridines. They bind at a site on the outer surface of the subunit. These drugs don't interfere directly with calcium flux, but act allosterically, in that they cause a conformational or shape change in the protein that holds the pore in a closed state. This symbol represents diltiazem, with both heart and vascular actions. The diltiazem binding site shows some overlap with the verapamil site and is also inside the pore, on the cytoplasmic side of the selectivity filter. Therefore, diltiazem can also physically impede calcium ion flux through the channel. It's not a pure pore blocker though. It has an additional allosteric action like the dihydropyridines to promote channel closure. Notice that the binding sites for diltiazem and the dihydropyridines lie within the transmembrane region of the channel. This means that they can sense and be affected by the membrane potential. In fact, both types of drug show an increased affinity for their binding sites when the membrane is depolarized. These differences in binding sites can help to explain the cardioselectivity of verapamil, the vascular selectivity of dihydropyridines, and why diltiazem has actions of both the other drug classes. Let's consider the allosteric inhibition of L-type calcium channels. Muscle cells of the heart and arteries have very different resting membrane potentials. Cardiac cells have a very negative membrane potential and depolarize for only brief periods during an action potential. This prevents allosteric inhibition by calcium channel blockers. In contrast, vascular smooth muscle cells are substantially more depolarized, even when they are not stimulated. This promotes allosteric inhibition and explains why dihydropyridines and diltiazem cause vasodilation and are effective in the treatment of hypertension. More than that, the different resting membrane potentials between cardiac and vascular muscle explains why dihydropyridines cause vasodilation without depressing the heart. The heart beats when action potentials arise in the sinoatrial node and spread through the atria to the atrioventricular node. There is a delay of around 100 milliseconds in the atrioventricular node before the action potential spread along conducting fibres to the ventricular muscle. In supraventricular tachycardia, the atrial or sinoatrial node cells fire action potentials at an abnormally high rate. If all these signals are transmitted to the ventricles, they contract at an abnormally fast rate, and there is insufficient time between beats for them to fill properly with blood. That impairs cardiac output. The aim of treatment is to prevent the excessive activity from reaching the ventricles. It can be done by slowing action potential conduction through the atrioventricular node. This gives the ventricles more time to fill with blood between each heartbeat, which improves cardiac output. The pore-forming action of verapamil and diltiazem on L-type calcium channels enables them to do this. The action potential of atrioventricular node cells is typical of a cardiac pacemaker. The membrane potential gradually depolarizes to a threshold at which L-type calcium channels open to mediate the upstroke. This differentiates atrioventricular node cells from the muscle cells of the heart, which use voltage-gated sodium channels to generate the upstroke. Verapamil can only access its binding site inside the channel pore when the channel is open. That occurs only during the active phase of the action potential. In between action potentials, the channels are closed. There is therefore only a brief period when verapamil can enter the channel pore and block the channel. It results in slowing of the upstroke and a reduction in amplitude, with the size of the effect increasing with drug concentration. An important property of drugs that block by binding in the pore region of a channel is that the block they produce is sensitive to the frequency of action potential firing. Let's look at a sequence of repetitively firing action potentials. They all have the same amplitude. If we now add verapamil, the next action potential is not much affected. However, as more action potentials fire, they become more blocked. That's because each action potential opens more calcium channels, allowing more drug to get into the binding site and exert its blocking effect. This is known as use-dependent block, because the more the action potentials open or use the channels, the more they become blocked. If the same experiment is repeated using the same concentration of verapamil but at a higher frequency of action potential firing, even more block develops. That is because the shorter time between each action potential means that the L-type channels are open for an even greater proportion of the time, allowing more block to accumulate. This is an important property. It means that L-type channel pore blockers, like verapamil, preferentially exert their antiarrhythmic effect during periods of increased activity. To sum up what I've covered, the cardiovascular calcium channel blockers act on the Cav1.2 channel subunit. Verapamil is cardioselective because it acts as a pore blocker. That gives it antiarrhythmic properties, making it a useful treatment for supraventricular tachycardias. The dihydopyridines are allosteric blockers and cause vasodilation without depressing the heart. Diltiazem is both a pore blocker and an allosteric inhibitor and is therefore both a vasodilator and a cardiodepressant. Due to their actions and effects, dihydropyridines and diltiazem are both effective in the treatment of hypertension and angina. The choice of drug would depend on the presence or absence of other comorbidities. I said at the start that amlodipine shows selectivity for cerebral arteries, but I've not explained why. Like the other dihydropyridines, it is an allosteric blocker. It is not yet known why it shows this selectivity, but it could reflect differences in the subunit composition of L-type calcium channels in cerebral arteries. Finally, gabapentanoids are a separate group of drugs that interact with the alpha-2 delta subunit to block trafficking of channel proteins to the membrane. With their selectivity for calcium channels in neurons, they provide effective treatments for epilepsy and neuropathic pain.